Welcome to Unit 3 in Summer School Biology I. This unit will be going over the big ideas of the organization of cell cycle and cancer, DNA structure and function, and protein synthesis. You will be investigating each of these topics for mastery of the concepts in this, and to use these concepts in the later work of the course. What will you be doing? 1. Cell cycle and cancer. The cell cycle can be thought of as the life cycle of a cell. In other words, it is the series of growth and development steps a cell undergoes between its birth, formation by the division of a mother cell, and reproduction, division to make 2 new daughter cells. To divide, a cell must complete several important tasks. It must grow, copy its genetic material, DNA, and physically split into 2 daughter cells. Cells perform these tasks in an organized, predictable series of steps that make up the cell cycle. The cell cycle is a cycle rather than a linear pathway, because at the end of each go-round, the 2 daughter cells can start the exact same process over again from the beginning. In eukaryotic cells or cells with a nucleus, the stages of the cell cycle are divided into 2 major phases, interphase and the mitotic, M phase. During interphase, the cell grows and makes a copy of its DNA. During the mitotic M phase, the cell separates its DNA into 2 sets and divides its cytoplasm, forming 2 new cells. Does cell cycle control matter? If you ask an oncologist, a doctor who treats cancer patients, she or he will likely answer with a resounding yes. Cancer is basically a disease of uncontrolled cell division. Its development and progression are usually linked to a series of changes in the activity of cell cycle regulators. For example, inhibitors of the cell cycle keep cells from dividing when conditions aren't right. So too little activity of these inhibitors can promote cancer. Similarly, positive regulators of cell division can lead to cancer if they are too active. In most cases, these changes in activity are due to mutations in the genes that encode cell cycle regulator proteins. Here we'll look in more detail at what's wrong with cancer cells. We'll also see how abnormal forms of cell cycle regulators can contribute to cancer. 2. Mitosis. Mitosis is a type of cell division in which one cell, the mother, divides to produce 2 new cells, the daughters, that are genetically identical to itself. In the context of the cell cycle, mitosis is the part of the division process in which the DNA of the cell's nucleus is split into 2 equal sets of chromosomes. The great majority of the cell divisions that happen in your body involve mitosis. During development and growth, mitosis populates an organism's body with cells, and throughout an organism's life, it replaces old, worn-out cells with new ones. For single-celled eukaryotes like yeast, mitotic divisions are actually a form of reproduction, adding new individuals to the population. In all of these cases, the goal of mitosis is to make sure that each daughter cell gets a perfect, full set of chromosomes. Cells with too few or too many chromosomes usually don't function well. They may not survive, or they may even cause cancer. So when cells undergo mitosis, they don't just divide their DNA at random and toss it into piles for the 2 daughter cells. Instead, they split up their duplicated chromosomes in a carefully organized series of steps. 3. DNA. Structure and replication. Term. Meaning DNA. Deoxyribonucleic acid. Nucleic acid that transmits genetic information from parent to offspring and codes for the production of proteins. Nucleotide, building block of nucleic acids. Double helix, structure of 2 strands, intertwining around an axis like a twisted ladder DNA replication. Process during which a double-stranded DNA molecule is copied to produce 2 identical DNA molecules, base pairing principle in which the nitrogenous bases of the DNA molecules bond with one another. DNA structure. DNA is a nucleotide, one of the 4 major groups of biological macromolecules. All nucleic acids are made up of nucleotides. In DNA, each nucleotide is made up of 3 parts, a 5-carbon sugar called deoxyribose, a phosphate group, and a nitrogenous base. DNA uses 4 kinds of nitrogenous bases: adenine, A, guanine, G, cytosine, C, and thymine, T. RNA nucleotides may also contain adenine, guanine, and cytosine bases, but instead of thymine, they have another base called uracil, U. Chargaff's rules. In the 1950s, a biochemist named Erwin Chargaff discovered that the amounts of the nitrogenous bases, A, T, C, and G, were not found in equal quantities. However, the amount of A always equaled the amount of T, and the amount of C always equaled the amount of G. These findings turned out to be crucial to uncovering the model of the DNA double helix. Double helix. The discovery of DNA's structure being a double helix was made thanks to a number of scientists in the 1950s. Image of a DNA double helix, illustrating its right-handed structure. The major groove is a wider gap that spirals up the length of the molecule, while the minor groove is a smaller gap that runs in parallel to the major groove. The base pairs are found in the center of the helix, while the sugar-phosphate backbones run along the outside. DNA double helix. Image modified from OpenStax CC by 3.0. DNA molecules have an anti-parallel structure. That is, the two strands of the helix run in opposite directions of one another. Each strand has a 5'd end and a 3'd end. Solving the structure of DNA was one of the great scientific achievements of the century. Knowing the structure of DNA unlocked the door to understanding many aspects of DNA's function, such as how it is copied and how the information it carries can be used to produce proteins. DNA replication. Semi-conservative replication produces two helices that contain one old and one new DNA strand. Semi-conservative replication produces two helices that contain one old and one new DNA strand. Semi-conservative replication. Image modified from OpenStax CC by 3.0. DNA replication is semi-conservative. This means that each of the two strands in double-stranded DNA acts as a template to produce two new strands. Replication relies on complementary base pairing. That is the principle explained by Chargaff's rules. Adenine, A, always bonds with thymine, T, and cytosine. C always bonds with guanine, G. Schematic of Watson and Crick's basic model of DNA replication. 1. DNA double helix. 2. Hydrogen bonds break and helix opens. 3. Each strand of DNA acts as a template for synthesis of a new complementary strand. 4. Replication produces two identical DNA double helices, each with one new and one old strand. DNA replication occurs through the help of several enzymes. These enzymes unzip DNA molecules by breaking the hydrogen bonds that hold the two strands together. Each strand then serves as a template for a new complementary strand to be created. Complementary bases attach to one another, AT and CG, DNA template strand, and the creation of its complementary strand, DNA template strand, and the creation of its complementary strand. The primary enzyme involved in this is DNA polymerase, which joins nucleotides to synthesize the new complementary strand. DNA polymerase also proofreads each new DNA strand to make sure that there are no errors. Leading and lagging strands DNA is made differently on the two strands at a replication fork. One new strand, the leading strand, runs 5 to 3 towards the fork and is made continuously. The other, the lagging strand, runs 5 to 3 away from the fork and is made in small pieces called Okazaki fragments. Diagram of leading and lagging replication strands. Diagram of leading and lagging replication strand. Example. Determining a complementary strand DNA is only synthesized in the 5 to 3 direction. You can determine the sequence of a complementary strand if you are given the sequence of the template strand. For instance, if you know that the sequence of one strand is 5 or AATGCC3, the complementary strand must have the sequence 3 TTACGG5. This allows each base to match up with its partner. ATTGGGC3, 3 TTAACCGG5. These two strands are complementary, with each base in one sticking to its partner on the other. The AT pairs are connected by 2 hydrogen bonds, while the GC pairs are connected by 3 hydrogen bonds. 5E, ATs 3 TTACGT5. These two strands are complementary, with each base in one sticking to its partner on the other. The AT pairs are connected by 2 hydrogen bonds, while the GC pairs are connected by 3 hydrogen bonds. Common mistakes and misconceptions. DNA replication is not the same as cell division. Replication occurs before cell division, during the S phase of the cell cycle. However, replication only concerns the production of new DNA strands, not of new cells. Some people think that in the leading strand, DNA is synthesized in the 5:3 direction, while in lagging strand, DNA is synthesized in the 3:5 direction. This is not the case. DNA polymerase only synthesizes DNA in the 5:3 direction only. The difference between the leading and lagging strands is that the leading strand is formed towards replication fork, while the lagging strand is formed away from replication fork. Khan Academy. 4. Protein synthesis. Term. Meaning RNA, ribonucleic acid. Single-stranded nucleic acid that carries out the instructions coded in DNA's central dogma of biology. The process by which the information in genes flows into proteins. DNA, RNA, protein polypeptide. A chain of amino acids. Codon. A sequence of 3 nucleotides that corresponds with a specific amino acid or start-stop signal during translation. Transcription process, during which a DNA sequence of a gene is copied to make an RNA molecule. Translation process, during which an mRNA molecule is used to assemble amino acids into polypeptide chains. Mutation, a change in a genetic sequence. Structure of RNA. DNA alone cannot account for the expression of genes. RNA is needed to help carry out the instructions in DNA. Like DNA, RNA is made up of nucleotide consisting of a 5-carbon sugar ribose, a phosphate group, and a nitrogenous base. However, there are 3 main differences between DNA and RNA. RNA uses the sugar ribose instead of deoxyribose. RNA is generally single-stranded instead of double-stranded. RNA contains uracil in place of thymine. These differences help enzymes in the cell to distinguish DNA from RNA. Image comparing the structure of single-stranded RNA with double-stranded DNA. Comparison of RNA and DNA molecules. Image modified from Wikimedia. CC BISA 3.0. Types of RNA. Type role messenger RNA, mRNA. Carries information from DNA in the nucleus to ribosomes in the cytoplasm. Ribosomal RNA, rRNA. Structural component of ribosomes. Transfer RNA, tRNA. Carries amino acids to the ribosome during translation to help build an amino acid chain. The central dogma. A gene that encodes a polypeptide is expressed in 2 steps. In this process, information flows from DNA, RNA, R protein, a directional relationship known as the central dogma of molecular biology, the genetic code. The first step in decoding genetic messages is transcription, during which a nucleotide sequence is copied from DNA to RNA. The next step is to join amino acids together to form a protein. The order in which amino acids are joined together determine the shape, properties, and function of a protein. The 4 bases of RNA form a language with just 4 nucleotide bases: adenine, A, cytosine, C, guanine, G, and uracil, U. The genetic code is read in 3 base words called codons. Each codon corresponds to a single amino acid, or signals the starting and stopping points of a sequence. Genetic code table. Each 3-letter sequence of mRNA nucleotides corresponds to a specific amino acid, or to a stop codon. UGA, UAA, and UAG are stop codons. AUG is the codon for methionine and is also the start codon. Genetic code table. Each 3-letter sequence of mRNA nucleotides corresponds to a specific amino acid or to a stop codon. UGA, UAA, and UAG are stop codons. AUG is the codon for methionine and is also the start codon. Transcription and translation. In transcription, a DNA sequence is rewritten, or transcribed, into a similar RNA alphabet. In eukaryotes, the RNA molecule must undergo processing to become a mature messenger RNA, mRNA. In translation, the sequence of the mRNA is decoded to specify the amino acid sequence of a polypeptide. The name translation reflects that the nucleotide sequence of the mRNA sequence must be translated into a completely different language of amino acids.